Hey AP, hope you're doing well when you watch this video. Thank you for watching, although I'm sure some of you felt like you didn't have a choice because it's an assignment, but nonetheless, I want to thank you. So yeah, we're going to talk today about biotechnology. It's not going to be any sort of intense heavy lifting, but definitely wanted to assign this video just because we're a little bit behind. We're trying to catch up, and this is pretty, for the most part, straightforward stuff, and we will review the next time we meet. So yeah, we're looking at two bacteria. These bacteria were actually bioengineered, which means they were created in a lab. And they were created in a lab to do one thing, and they were created in a lab to clean up oil. I'm not sure if you saw the movie Deepwater Horizon, but that oil spill that happened many years ago, it was devastating to the ecosystems. But what if we could bioengineer? What if we could engineer species like bacteria to clean up the oil? But what are the side effects? What are the consequences of that? You know, if we release them, could they change the ecosystems? And so it's not just, this is called bioremediation, solving problems using life. But it's not just that simple, right? For every action, there's a consequence, right? And so we're just wondering, what that is. An example from your book is dental disease in dogs. Toxins can be absorbed into the bloodstream, cause infections, damage organs. And research has modified tobacco plants to produce vaccines against this. So biotechnology can be used to modify bacteria to even make insulin or eat pollutants like oil. And so we're going to talk about it. So how is biotechnology used to alleviate environmental problems? That's a big question. Here's your objectives from AP Classroom. I gave you the questions, of course, with this assignment to turn in. But this is something that I want you to definitely pause and read over because this is directly from AP Classroom. Okay, so let's go ahead and get started. Cloning. Cloning is the production, DNA cloning specific, is identical copies of DNA cells or organisms. Of course, the first animal to be cloned was Dolly. Identical twins are clones, but we can actually clone DNA. So gene cloning is the production of many identical copies of the same gene. So we could technically start treating people by replacing genes, replacing their DNA. Obviously, there's a lot of question marks to this. How does it work? Is it safe? The first person to actually undergo gene therapy, I forget his name, but he tragically passed away. It was many years ago, but it's definitely developing science, treating people with correct genes. And so complementary base pairing of DNA is the basis for nucleic acid hybridization. The base pairing of one strand to a complementary strand. So A, T, C's, and G's, right? But because of that, we can exchange DNA with other species and create recombinant forms. This part of DNA is from one species and this part is from another species. And that leads us to genetic engineering, the direct manipulation of genes for practical, useful purposes. Biotechnology began with the use of organisms to produce products such as alcohol and acetone. Now it's possible to even modify organisms with genes from other distantly related organisms. So this is called, and I would like this one of your questions, recombinant DNA. Recombinant DNA is DNA molecule made in the laboratory that is derived from two or more sources. And there's many types of that. If you click on that link, example would be glowing fish in aquariums and different types of transgenic animals. And so let's start talking about bacteria. Bacteria contain plasmids. So plasmids are small circular DNA molecules that is extra from the chromosome. Okay, so this is like extra DNA inside of the cell. And so to clone a piece of DNA using bacteria, researchers first obtain a plasmid and insert the DNA from another source or foreign DNA. And that is called recombinant DNA. And so this, of course, has revolutionized biotechnology, and this has developed biotechnology. And biotechnology is the manipulation of organisms or their genetic makeup to make, again, useful products. So here's an example of that. Bacteria, plasmid, here's the plasmid, here's the genome of the bacteria, here's the plasmid. And so let's say we have a gene of interest that we want to make. Well, we could splice that gene out, insert it into the plasmid, put the correct promoters and all sorts of various stuff with it. And you can see that plasma is now put into the bacterium, and that's called a recombinant plasmid. And so that gene could be used to make pest resistance. Gene could to alter bacteria for cleaning up toxic waste, which we already talked about. We could actually produce hormones as well, which we do. We produce insulin and different hormones because we harvest the protein that the bacteria makes, purify it, and use it as medication, which has completely revolutionized treating some disorders and diseases. And so again, this is called gene cloning. Okay, and we're trying to produce a protein product. What do we require to do this? Well, we require a vector. A vector introduces that recombinant DNA into the host cell. So plasmids, these are common vectors that we use. Also, we could use, sometimes they use viruses and various things, but two enzymes are required to introduce DNA. A restriction enzyme, which we're about to talk about, and DNA ligase. Ligase is the gluer, and so DNA ligase is the enzyme that seals DNA into an opening created by the restriction enzyme. So this is the gluer, seals it all together. Restriction enzymes cut it in specific places. So bacterial restriction enzymes cut DNA. This is kind of like a defensive enzyme. You know, when they were attacked by bacteriophages, they would use these enzymes to break down the DNA to protect themselves. But we actually, scientists, over many years, have actually used this DNA to develop, you know, these products, these protein products and gene products. And so restriction enzymes cut DNA at specific places, which we'll talk more about. They can create blunt ends or sticky ends, which we'll talk about again here in a second. And DNA ligase closes it up. So here's a picture. So you have restriction enzymes, which cut DNA at specific places, create these sticky ends, which means they can join together. A DNA fragment from another source is added, again, cut with a restriction enzyme. And now we put it together, you know, put them all in the same place, and use DNA ligase to join and seal the strands. And that's your recombinant DNA. Here's a picture, I believe, from your textbook. You have human DNA, bacteria, again, DNA, A, T, C's, and G's, right? Both for the human and the bacterium. And so DNA ligase seals it, and we have gene products. We make products. We make insulin, which is, of course, helps people who have diabetes, and that we harvest those products. And so here is restriction enzymes. They cut that bond right there between the sugar and phosphate in DNA. And so they used it as protection, right? Bacteriophage injects the DNA, tries to take over the host cell and infect it. Bacteria use these enzymes to protect against that. They cut it into pieces, and that DNA possibly no longer is incorporated. And so there's different ones. You don't have to have them memorized, you know, BAM-HI, HIND3, EcoRI. You don't have to have these memorized, but just know that they cut at the same place. So you see GAATTC cuts between the G and the A on the 5' end. Well, it cuts over here at the same place. And so that is how it cuts. So it's, think kind of like race car. Restriction enzymes have two identical active sites. They cut on both strands. And so race car, if you were to cut between the race car is the same in both ways. And so that's kind of how DNA, so it cuts in both directions. Okay, and so sticky ends is ones that we would like to have because we want to use those as complementary sequences. So for example, you know, blunt ends, we couldn't join blunt ends together with anything. But sticky ends you could join. Sticky ends you could take and join them together, okay? And then you create like these like recombinant DNA structures. And so here's just another picture here. EcoRI cuts at the red arrows. We can join it together using DNA ligase and creating that. And so key tools for manipulating DNA, restriction enzymes for cutting DNA into fragments, ligase for joining, and then let's talk a little bit about Pcr and gel electrophoresis now. Pcr amplifies DNA sequences. So we might just have a little DNA, but the polymerase chain reaction literally lets us amplify those amounts of DNA, which is extremely helpful because, of course, we need as much DNA as possible to probably pull all this off. Gel electrophoresis analyzes it and purifies it. Hopefully we get that crime scene. We will actually use gel electrophoresis in lab. And so the polymerase chain reaction produces many copies of a specific target segment of DNA. And so it amplifies it. So during each PCR cycle, the mixture is heated to separate DNA. It's cooled to allow the short-stranded DNA primers to complementary base pair. And then DNA polymerase, the builder, right? We build all these more molecules of DNA. So it kind of looks like this. Denature it, allow it to cool to let the primers join, and then we let it build, and then we repeat it, and we repeat it, and we repeat it, and all of a sudden we have greatly enhanced the DNA that was there. And so, of course, you could probably think about this as practical usage, but let's say we just find a little bit of DNA at a crime scene. Well, we can use PCR to greatly amplify that to do multiple crime scene investigations, genetically speaking. And so PCR amplification is used to provide the specific DNA fragment to be cloned. So here's just another picture of that DNA fragment obtained by PCR, cut by the same restriction enzyme, right? Mix and ligate, and there you go, recombinant DNA. Applications of PCR, they're short tandem repeat profiling. So these profiling techniques, unique collection of different fragments is produced, and then gel electrophoresis, which we'll talk about next, separates the fragments according to charge and size. Remember, DNA is negatively charged, so it goes towards the positive side. But the longer pieces take longer to move through. Basically, it's like an agarose. It's like a maze of agarose. And I'll show you a picture here in a second. And they basically work their way through based upon positive and negative attraction, and the short pieces go further in the time allotted than the longer pieces. And it produces a distinctive band pattern called DNA fingerprint, or it's also called DNA profiling. This is used in maternity suits, rape cases, corpse ID, of course, all this justice that we need to carry out, to love justice. God calls us to love justice. And so here's an example here, right? So who's the father? Well, here's the mother's, and here's the child's. So you can see that right here, boom, boom, male 1 would be the father. And so that's called gel electrophoresis, which is right here. So you can see that DNA moves this way, positive, negative, cathode and anode. DNA is negatively charged. And, excuse me. And so you can see that here's standard links, here's restriction fragments of unknown links, restricted fragments of standard links, and we can figure out how big these pieces are, or we could actually even figure out suspects or all sorts of things. And this is DNA gel electrophoresis, which I hope to do in class with you as well. And so hundreds of restriction enzymes have been purified and used, and these are, of course, separated by size. So GMOs, what are GMOs? GMOs are genetically modified organisms, or genetically engineered, and they can produce biotechnology products. And so this is a huge thing, because I don't know if you ever shopped with anyone at a grocery store. I'm sure you have. I don't know if you look at labels or you just kind of zone out. But yeah, GMOs are definitely a thing. Are they healthy to use? Are they not healthy to eat? Non-GMO products are popular, all natural. And so recombinant DNA, these altered host cells, and transformation, these genes are inserted into host bacterial cells, and this is called a transgenic organism. So GMOs are transgenic organisms. So an example of this is, so this is a genetically modified organism. So here's a foreign gene inserted in. You have your promoter region, transcription, and then this foreign gene is located in there, and the foreign gene is expressed because gene expression for the vector is present. So here's some medically useful products of bioengineering: erythropoietin, human growth hormone, insulin, factors which help blood clot correctly, a tissue plasmid activator which dissolves blood clots after heart attacks or strokes. If they think you're having a stroke, they'll give you that very, very quickly. So here's insulin. So you have insulin product being made, 2 of them together, purify it, join them together, there's your active insulin, okay? And so, of course, we want to make sure it's ready to be used by humans. So transgenic bacteria, we've already talked about it a lot. Products include, even vaccines, hormones. Transgenic bacteria can produce chemical products. Bioremediation, we talked about that. Like, for example, transgenic bacteria can produce frost-resistant strawberries. But is it healthy? Is it right? There's a lot of, kind of debate out there. Oiling bacteria, etc., so on and so forth. We've actually, genes been introduced into plants, immature plant embryos, or plant cells. And so now cotton, corn, soy, potatoes, all these are genetically modified. Soybeans are now resistant to a common herbicide. So basically you can spray a different pesticide on it and it doesn't die, it lives. So of course that increases crop yields and products, but is it healthy? Is it right? Is it wrong? Some people like to debate that stuff. Human hormones, of course. Genes can be inserted into eggs of animals. This is called farming. And so eggs can be placed in an agitator, and the needles make tiny little holes, and we can insert DNA from that, called transgenic organisms. So this is called farming, production of pharmaceuticals, for example, in farm animals. So for example, transgenes are inserted next to the promoter for lactoglobulin, a protein for milk. So the transgenic animal then produces large quantities of the protein in its milk. So you make this, you put whatever gene you want, and then so what would it be like to be on a farm and you're milking the dairy cow, but instead of just milk, you're having a protein that you need, or you have a medication, and you could purify that and have your medication just by milking a cow and purifying it, of course. And so, of course, therapeutic uses. Plans are to produce drugs that treat cystic fibrosis, cancer, blood disorders in the milk of an animal, okay? So here's kind of what we're talking about there. You can pause and kind of look at this picture. But that is crazy to think about, that you can literally purify milk from an animal to get a drug. Here's a human growth hormone deficiency, lacking in growth hormone. So human growth hormone can now be produced by transgenic cows. So here's the crazy thing. According to a textbook I have, with only 15 such cows, we can supply HGH for all the children in the world that suffer from pituitary dwarfism, which is insane to think about. That's just insane to think about, that we could treat diseases by modifying animals and making the drug in their milk. Crazy to think about. Okay, and here's just an example of other potential applications of biotechnology. Here's just some specific examples that I have from another textbook. The bacterium Bacillus thuringiensis, produces a protein Bt that kills insect larvae. So we've injected this toxin into plants, and so the pesticide use is reduced because now the plants just make this toxin. It has been shown to be safe as far as it doesn't hurt animals like us to eat. Vitamin A is very, very common deficiency in certain developing parts of the world. But most people every day eat rice and beans. What if we could grow rice with vitamins in it? And so this is what they do. Wild type, here's golden rice 1, golden rice 2. This is rich in beta carotene, which is converted vitamin A in the body. So when you're eating a bowl of rice and beans, you're eating vitamins. That's amazing. Plants that are salt tolerant. So crops are environmental conditions, right? Some soil is very high in salt near the ocean, various reasons. What if we could do that? What if we could cause a plant not to look like this when it's grown in salty soil, but it thrives. It thrives, and that's a transgenic organism, okay? So there are some concerns about genetically modified crops. So for example, it's unnatural, okay? Nature has done it well for thousands of years. Why interfere? Genetically altered foods are unsafe to eat. Are they? Genetically altered crops are dangerous to the environment. What if they got out? What if they got into the wild population? Would they completely change ecosystems? Advocates point out that all crops have been manipulated. So GMOs, we've been eating those for years and years and years because we've been manipulating them through agricultural practices and fertilization and cross fertilization. Single genes for plant are inserted. Advocates contend they are safe. And concern over environmental centers on escape. So if a gene were transferred to a closely related weed species, centers on escape, which would be very unlikely. So widespread use of glyphosate has resulted in selection of gene mutations. So more than 10 resistant weeds. There's a lot of resistant weeds out there. Natural selection, we'll talk about it. And so usually only a few cells are exposed to recombinant DNA are actually transformed. So it doesn't just mean, so here's the last thing we'll kind of talk about today. It doesn't mean like once you just insert a gene, that gene's like, oh yeah, we're just going to take it up and we're going to start making it. No. So in order to understand if the recombinant DNA has actually been taken up, recombinant DNA includes selectable marker genes or reporter genes, such as P-glow. P-glow actually makes things glow in the dark. It's crazy to think about. Here's bacteria glowing in the dark. Here's mice glowing in the dark. These are genetically modified mice. And so about like 1 cell in 10,000. So we need to know if it has been taken up. And so these are called selectable markers. We use antibiotic-resistant genes, so we grow it on a petri dish full of the antibiotic. Whichever cells survive, we know they took up the recombinant DNA. Only cells carrying the resistant gene can grow in that part of the body, on the antibiotic. And so a reporter gene is one that codes for a product that's easy to measure. So glowing in the dark, growing on an antibiotic-resistant place, that's kind of where it's at. And so here's an example here. A plasmid has genes for ampicillin resistance and beta-galactidase, which turns colonies blue. White colonies have an inactivated gene. And so we're trying to insert the DNA into the restriction site. So here's the restriction site. It's within this gene. Here's the foreign DNA. After cutting the plasmid, how do we know it's there? Well, host bacteria are transformed with the recombinant DNA and grown on that medium with ampicillin resistant and the substrate. White colonies have an inactivated LAC-Z gene, indicating that they carry the recombinant DNA. Blue colonies have the intact LAC-Z. So LAC-Z, beta-galactidase, if they turn blue, this recombinant DNA didn't go in. And so that's just an easy way of measuring that stuff. Of course, glowing in the dark comes from a jellyfish. Major goal molecular cloning experiments also beneficial is to try to figure out what DNA leads to and what proteins consequences they are, okay? We already talked about bioremediation. And so the last thing really quick, woolly mammoth. They're trying to bring it back. I don't know if you watched Ice Age or not, but it's it's a show. And so this is called, in order to do this, we have to sequence. So once a gene is cloned, complement-based pairing can be exploited to determine the gene's complete nucleotide sequence. This process is called DNA sequencing. So we need to be able to sequence DNA to be able to figure out what the future DNA is, the rest of the DNA. And so they sequence DNA by synthesizing the complementary strand of a single immobilized template strand. You don't have to understand how this happens for sure, but this just kind of shows you that it's important for DNA sequencing to happen. It's important that it is there for that reason. And so one really amazing thing is CRISPR. You can look up CRISPR. I'm not going to dive into it right now with you, but it's basically this huge, powerful new technique where CRISPR basically cuts out strands of DNA and replaces it with other DNA. And we can knock out genes to study. We can also replace DNA. There's a lot of applications to CRISPR, and I'm not going to get into it because it's just beyond the scope of this course, but nonetheless. Okay, so there's some questions there. So of course you have the questions in the email that I emailed out. Thank you so much for watching. If you watched this far, I'm literally so proud of you. It's amazing. And you can tell me this word in class and I'll give you some extra credit, okay? And don't tell anyone else. If you watch this far, don't tell anyone else. But what phrase am I about to say? And I'm about to say God is good all the time. And if you write that on the test, don't tell anyone, but if you write that on the test, God is good all the time, I'll give you a little extra credit for watching this far. Thank you so much. Hope you're doing well, you and your family. Have a great rest of your day.